Okay, have you ever just stopped and wondered, could we actually change the fundamental building blocks of life? I mean, to literally go in and edit the code that makes a plant or an animal an animal? Well, let's get right into it, because this isn't some far-off dream anymore. We're actually doing it.
I mean, it sounds like something straight out of a sci-fi movie, doesn't it? A fish that glows in the dark, like a living nightlight. But seriously, just think about that for a second. Could you really take a trait from one creature, like a glowing jellyfish, and just give it to something else entirely?
Well, surprise, the answer is yes. We already have. This is the glowfish. What scientists did was they took the gene that makes jellyfish glow, that bioluminescence, and they popped it right into a zebrafish's DNA. It's a perfect glowing example of what genetic engineering can do. This isn't science fiction anymore, folks. It's happening right now.
Okay, so how on earth does this all work? How do we actually edit the code of life? Let's break it down.
So, at its very core, genetic engineering is all about directly manipulating an organism's DNA to get a specific result. I want you to think of it like editing a document on your computer. You can go in, delete a sentence, maybe add a new one, or even just change a few words, and boom, you get a totally different outcome. In this case, a desired characteristic.
Now, to do this kind of high-tech editing, you obviously need the right tools. And believe me, over the last 50 years or so, scientists have developed a pretty incredible toolkit for messing with genes.
And this toolkit has evolved a lot. The original method was called recombinant DNA, and you can think of it as the copy and paste of genetics. But the new kid on the block, CRISPR, is a total game changer. It's more like find and replace. Way faster, way more precise. It has absolutely revolutionized the entire field.
All right, so let's dig into that original copy and paste method. First, scientists find the gene they want. Let's say the one for human insulin. Then they take these things called molecular scissors and snip open a little loop of bacterial DNA called a plasmid. Then they just paste the human gene right in. They put this new combo DNA back into a host cell, and that cell basically becomes a tiny little factory, just pumping out pure human insulin.
And the result of this, well, it completely changed how we treat diabetes. I mean, before this, insulin had to be taken from animals, which, could cause all sorts of allergic reactions. This new synthetic insulin that came out in the late 70s was safer, way more consistent, honestly, a landmark achievement that has saved millions and millions of lives.
Okay, now for the new find and replace tool, CRISPR. This is where things get wow, incredibly precise. You have this thing called a guide RNA that basically acts like a tiny GPS. It leads an enzyme, Cas9, to the exact spot on the DNA strand. Then, snip, Cas9 makes a perfect cut. This kind of accuracy means scientists can now remove, replace, or add genes way more easily and cheaply than ever. And that, right there, is why everyone calls it a game changer.
So, with these super powerful tools in hand, what have we actually created? Well, the applications are already reshaping our world, everywhere from our farms to our pharmacies.
Okay, let's take a look. One of the very first genetically modified foods to hit the shelves way back in the 90s was the Flavor Saver tomato. It was engineered to ripen much more slowly, so it would stay fresh on the shelf for longer. It was really our first little glimpse into the future of food.
And then things got even more interesting. We started making crops that could protect themselves. Take BT corn, for instance. It actually produces its own natural insecticide, thanks to a gene from a soil bacterium. And what does that mean? It means farmers can use way fewer chemical pesticides. That's a huge potential win for the environment.
We can also use this tech to pack more nutrition into our food. This right here is golden rice. It's been modified to produce beta-carotene, which our bodies convert into vitamin A. Now, in parts of the world where vitamin A deficiency is a massive problem, causing blindness and even death, a food like this could be an absolute lifesaver.
And hey, it's not all about solving these huge, serious problems. Sometimes genetic engineering is used for pure aesthetics, to create novel things that just couldn't exist in nature, like this incredible blue rose. Okay, so the potential is obviously incredible, but this is where we have to pause. Just because we can do all these things, does that mean we should? And that brings us to the other side of the coin, the perils, and the really tough questions we have to start asking. Because when we shift this conversation away from plants and over to people, well, the ethical stakes just skyrocket.
We're not just talking about cool science anymore, we're talking about the future of our own species.
So it all comes down to balancing the scales, right? On one side, you've got these incredible advantages: more food, amazing new medicines, more resilient crops. But on the other side, the risks are just as huge. We're talking about the potential to wreck entire ecosystems, run into unexpected health problems, and walk right into an ethical minefield.
Ultimately, where this technology goes depends on whether we, the public, accept it. But what actually shapes our opinion on something this big? It turns out it's a lot more complicated than just making a list of pros and cons. Get this, a really fascinating study from 2023 found that the single biggest thing that determines if we accept these technologies is the perceived benefit. It's not about the risk, it's not even about how much we trust the people in charge. It really boils down to us asking, okay, but what's in it for me? How does this actually help? If that benefit is clear and big enough, we're way more likely to say yes.
And these right here, these are the debates that are keeping scientists and ethicists up at night. You've got the whole designer baby sphere, the idea that we could engineer traits and create a huge genetic gap in society. Then there are the environmental risks we just don't know about yet. And maybe the biggest question of all, who gets to say yes to changes that will affect every single human generation from here on out?
So look, this technology, at the end of the day, isn't good or evil. It's just a tool. A really, really powerful tool. And like any tool, whether it builds something amazing or causes a disaster, depends entirely on how we decide to use it. So, the path forward has to be about finding that critical balance. We absolutely need to keep pushing the boundaries of what's possible, but we have to do it with a really strong sense of ethical responsibility for the world we're creating. For all of human history, we've only been able to read the book of life. Now, for the first time, we can pick up the pen and start writing in it ourselves. So the real question for all of us is, what story should we write? What kind of future are we going to create? That's the question we're all left with.